Okay, so hello students. Hope this video finds you guys in good health. Welcome to the lecture 5 of sexual reproduction flowering plants. Today, talking about the topics that we are targeting to complete today. It includes outbreeding devices, next pollen pistil interaction, followed by double fertilization. So I hope that by lecture 5, I am able to complete these topics so that in lecture 6, we can move on to post-fertilization events where we are going to study about endosperm, embryo, fruit, seed. Okay, so starting with outbreeding devices. Okay, so before I get into further details, the first two topics, outbreeding devices and pollen pistil interaction, they are more of analytical topics. Okay, so all the basic facts or information or conceptual understanding that you require to understand these topics, I have already covered them in the past 4 lectures. So if you have gone through the lectures and read the materials properly, then it won't be much tough for you. It's a bit of a more analytical portion. So we are going to complete for these two portions at least, we're going to completely stick to the NCERT. I'm going to literally read the NCERT line by line and make notes and make the concepts clear accordingly. In like for these two topics, outbreeding devices and pollen pistil interaction, we are going to keep NCERT in the essential center of our discussion. Okay, so yeah, so starting with it. So the first we have is pollen pistil interaction. Sorry, my pardon. First we have is outbreeding devices. Next we are going to discuss pollen pistil interactions. So going by the NCERT, NCERT says majority of flowering plants produce hermaphrodite flowers and pollen grains are likely to come in contact with the stigma of same flower. The first is what does hermaphrodite mean? So in easy language, you can say this to be bisexual. Okay, so we observe that majority of the flowers, they are bisexual in nature. That is, they have androecium and gynoecium both, the male and female reproductive holds. Okay. Okay, so now, as I already mentioned in the previous lecture, so if a flower is bisexual, it has a higher tendency to conduct autogamy, that is transfer of pollen grains from anther to stigma of same flower of the same plant. So like of the same flower, then it's obviously the same plant. So in cases where we observe bisexual flowers, there is a higher chance that the pollen grains are likely to come in contact with the stigma of the same flower. That is, we are referring to autogamy. So bisexual flowers, that is higher chance of autogamy. So next, continued self-pollination results in inbreeding depression. Okay, so I think I have missed this point in the last discussion. Self-pollination, cross-pollination. Self-pollination is easier. Obviously, you can understand the reasons. Self-pollination is easier because you see there is no Okay, let me just write this. Self-pollination, there is no pollinator involved. So this method of self-pollination is not dependent on any other agent. Okay, so we have a single flower, we have the male reproductive as well as the female reproductive whole in the same flower. So there is no pollinator involved. It is a more surer process. It is, and there is, and like if you consider the genetic aspect, genetically also the characters are conserved, right? Because here we are not observing mixing of characters because the male and the female reproductive whole, they're of the same flower. So genetically also, like there is no intermixing. Okay, so there is no pollinators involved. That's why it's a more surer and quicker process. Like it is involved in a single flower, right? And the third point is no genetic intermixing, because here we are talking of the same flower of the same plant. Now, cross-pollination is just the opposite of this. Okay, in the cross-pollination, pollinators are involved. We have studied about that in well detail. Like the pollinators include air, water, we have birds, we have insects, we have bats, snakes, snail. I'd covered that. So for cross-pollination, we have pollinators involved. It is also not a surer process, as in, like, see, whenever there is a third party involved in any process, its surety decreases. As in, like for example, consider an anemophilous flower, air pollinated flower. Okay, so the pollen grains are being released, is moving to the wind, but due to some natural calamity or natural, maybe the wind direction changes, or maybe there's a storm, the pollen grains land on a different place rather than the stigmatic surface where it's supposed to land. So pollen grain is a waste. So cross pollination is not a surer process. But the good thing about cross-pollination is there is genetic intermixing. So this is the highlight point. So cross-pollination, we have pollinator involved, not sure process, and I have described the reason as well. And the last is there is genetic intermixing. So what we observed in xenogamy, that pollen grains are being transferred from anther to stigma of different flower of different plant. So the genetic constituents are different, right? So when the genetic constituents are different and union is happening between the two, fertilization is happening between the two, there is genetic intermixing. Okay. So now here comes the concept of inbreeding depression. When there is repeated self-pollination, there is no genetic intermixing. And when there is no genetic intermixing for consecutive generation, this gradually leads to inbreeding depression, which is not at all desirable. When there is repeated self-pollination across generation, there is no genetic intermixing, it results in higher chance in the development of inbreeding depression, which is not at all a desirable phenomenon. Okay, so you're going to find the reference of this inbreeding depression more in the chapter strategies of plant and food management in that chapter. Okay, but here it is sufficient to understand. It would suffice to understand that continued self-pollination would result in inbreeding depression. The reason is very clear that there is no genetic intermixing for the consecutive generations. So flowering plants have developed many devices to discourage self-pollination and encourage cross-pollination. The reason, see, genetic intermixing. See, this is quite an interesting stuff, right? The cross-pollination is a more sure, cross-pollination in comparison to self-pollination is a less surer process. A third party, a pollinator is involved, right? In cross-pollination, there is a pollinator involved. It is a less surer process. It's more time taking. And self-pollination, no pollinator is involved. No third party is involved. It's a more surer and quicker process. In spite of that, the flowering plants have developed many devices to discourage self-pollination and encourage cross-pollination. The only reason being genetic intermixing. When there is no genetic intermixing for consequent generation, it leads to inbreeding depression, which is not at all desirable. So genetic intermixing is very important. It promotes variation. It promotes evolution as well. So now we are going to study about what are the different mechanisms that the flowering plants have taken up so that it can prevent self-pollination and promote cross-pollination. So the first point is, in some species, the pollen release and stigma receptivity are not synchronized. You have to remember, I have discussed this in the part contrivances of cross-pollination. So there is difference in the time of maturity of the male and female sex organs. So first I'm writing pollen release and stigma receptivity are not synchronized. Okay, so I have covered this in detail, the contrivances of self-pollination as well as cross-pollination. So it was a point of cross-pollination, that pollen release and stigma receptivity, that is the maturity of the male reproductive hole and the female reproductive hole, are not synchronous. So there we had observed two conditions. One is the proto-androus condition, where the male reproductive hole was maturing before the female, and the protogynous condition, where the gynoecium was maturing before the male, that is androecium. Okay. So the next point is, okay, so in some species, the first point was, in some species, the pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma, that is the proto-androus condition, before the receptive, or the stigma becomes receptive much before the release of the pollen. This is protogynous condition. We have discussed that along with the examples. Keep the examples in mind, they are important. Okay. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. We have also discussed that. Okay. So let me just write it. So we have our, okay. So what do we have here is, okay, let me just wait a second. Let me just open the slide that where I had discussed all this in detail. Okay, so here, this is dichogamy. You will find I have discussed this in detail in lecture number 4. What is dichogamy? The maturity of the sex organs at different times. So pollen release and stigma receptivity, they are not synchronized. That is, in one case, the anther is matured before the stigma style ovary, that is the gynoecium. And in the other case, the gynoecium is maturing before the androecium. Okay. So this is dichogamy. Next is heteromorphism. Heteromorphism, you have to remember the anther and stigma are placed at different places. And I had also mentioned that the transfer of pollen grains would occur between the anther and stigma who are at the same height. So if the structure is something like this, I'm drawing it in rough. And we have this structure. And consider that I have this a taller one and I have this a shorter one. So here we are going to observe pollen transfer from here to here and here to here. So pollination would occur among the same height. Okay. Now the third point is the third device to prevent inbreeding is self-incompatibility. Okay, so this is one. So I think you will clearly understand this outbreeding devices topic is kind of equivalent to contrivances of cross-pollination. So okay, I think this is looking more like gauze. Okay, it's cross-pollination. So see, there are a lot of it's essentially the same thing. Outbreeding devices, what is like, consider go by the definition. In outbreeding devices, the plant is adopting some mechanism so that it prevents self-pollination and promotes cross-pollination. That's the same as contrivances of cross-pollination. So all this stuff I've discussed in detail. Now, which I had not discussed in the previous lecture, it is self-incompatibility. Okay, so I am erasing this because you will find all this in very detail in lecture number 4. So let's discuss in more detail what I had not discussed before, that is self-incompatibility. Okay, maybe I had mentioned it somewhere in the past lectures, but today we are going to discuss that in very detail. Okay. What does the NCERT say? The third device to prevent inbreeding is self-incompatibility. This is the genetic mechanism and prevents self-pollen from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Okay, so what happens? This is a stigma, this is a style, this is the ovary. Okay, so now consider this is our one flower plant. So I really hope that you guys are keeping in good health because the weather is really harsh in this time of the year. Okay, so I hope you guys are doing well. Not like me, I'm not well currently. Okay, anyhow, so coming back to the lecture, see, what does self-incompatibility say? It is a genetic mechanism and prevents self-pollen. So you have to understand what is self-pollen. So I think it is pretty evident from the term itself, the pollen of the same plant. See, reading the NCERT, self-pollen from the same flower or other flowers of the same plant. So here self-pollen means the pollen grain of the same flower or different flower of the same plant. So we can encompassing both, we can write pollen of the same plant. So you have to remember we had autogamy, geitonogamy, and xenogamy. Autogamy and geitonogamy were self-pollination, mechanically, they were self-pollination. And in case of xenogamy, it was cross-pollination. And geitonogamy, in geitonogamy, we can say mechanically it is self-pollination because it is of the same plant. But since it is different flower, so we can observe variations. Okay. So what is self-pollen? The pollen of the same plant. So what is happening? Okay, so let me draw the diagram. So this is the pollen. So this pollen is falling on the stigmatic surface. So now let me just draw the enlarged view of this. For example, I'm taking this as my example. So I'm drawing the enlarged view of this. So we have the stigmatic surface. Pollen grains are falling like this. The pollens have this pollen kit outside. You have to remember. I think this was discussed in the first lecture. So breaking this pollen kit. No. Okay, let me just draw over this only. So breaking this pollen kit, the pollen tube is trying to develop. Okay, so pollination has happened. The pollen has fallen onto the stigmatic surface. Now, what does it say? This is a genetic mechanism which prevents the self-pollen, that is pollen from the same flower or of different flower of the same plant, from fertilizing the ovules. How do they do that? They inhibit pollen germination or pollen tube growth in the pistil. So, one point is pollen germination. What is pollen germination? It is clearly given in the NCERT. Pollen tube growth through the pistil. So how is self-incompatibility, preventing pollen germination. It is doing that by inhibiting the growth of pollen tube. For example, this is, for example, a self pollen. I'm writing it as self only. And maybe it's a non-self pollen. So what does it do? If it's a self pollen, so it is going to inhibit the formation of pollen tube. But if it's a non-self pollen, it's going to support, it's going to enhance the rate of growth of this pollen tube. Okay. So this thing comes under self-incompatibility. Okay. This is not self-incompatibility, obviously. So you go by the term self-incompatibility. It is incompatible to the self-pollen. So if self-pollen is falling onto the stigmatic surface, this is a genetic mechanism adopted by the flower which prevents the growth of pollen tube of the self-pollen, hence preventing pollen germination. And as pollen germination is prevented, the pollen cannot fertilize the ovary. Okay. So now coming to the fourth point, which is another device to prevent self-pollination, is the production of unisexual flowers. We have already discussed that if it's a unisexual flower, it has only one of the reproductive holes, either androecium or gynoecium. So essentially, it has to be cross-pollination with another flower so that we can obtain fertilization. Okay. So, okay. So here are some examples given in the NCERT. Let's just take a look at them. Okay. Okay. So we have castor and maize, and another example we have papaya. So let's just give it a think. Okay, so we have already discussed outbreeding devices. Outbreeding devices is essentially equal to contrivances of cross-pollination. In that, we had already discussed about dichogamy, herpogamy, heteromorphism. I was talking of hermaphrodite. Hermaphrodite means bisexual flowers. So production of bisexual flowers is a strategy, is a contrivance for self-pollination. But when you're talking of strategies for cross-pollination, is unisexuality, production of unisexual flowers, is dichogamy, herpogamy, and heteromorphism. And there was another point which I had not discussed back then, that is self-incompatibility. Okay, the self-incompatibility, by genetic mechanism, the flower is preventing the development of pollen tube from the self-pollen, thus preventing pollen germination. And there is another part, it's not like it doesn't come under self-incompatibility, but it also happens as a result of the chemical dialogue between the pollen and the stigmatic surface, and it promotes the growth of pollen tube of the non-self pollen. So now coming to the example part, now think of it, is autogamy, geitonogamy, xenogamy. So to prevent autogamy, autogamy is transfer of pollen grain from anther to stigma of same flower same flower, it has to be same flower. So to prevent autogamy, we are going to produce unisexual flowers. This is a clear strategy, right? So if you have unisexual flowers, the flower produced either has to be male or a female. So obviously autogamy can't happen. So now, but like consider the case of castor and maize. So it has male flowers or female flowers, but on the same plant. Okay. So what happens in castor and maize? So it has either male flower or female flower. That means in terms of flowers, it is dioecious. So it produces dioecious flowers. So it either produces male flowers or produces female flowers. So there are unisexual flowers. So autogamy is not an option in case of castor and maize. But geitonogamy is possible because male and female flowers are present in the same plant. Right. So transfer of pollen grains from anther to stigma of different flower but of the same plant, that is geitonogamy. It is possible. So geitonogamy is possible. So see, but male and female flowers, they're present on the same plant. So the plant is monoecious. So now think of it. The particular species of plants which produce monoecious plant, that is male and female flowers, both are present in the same plant, but produce dioecious flowers. Dioecious flowers means the flower either is male or is female. In such cases, autogamy is prevented, but not geitonogamy. Now, in case of papaya, it produces either male plant or produce female plant. So this is essentially the dioecious condition. So now, in case like either a papaya plant is either male plant or female plant. So there is no chance of either autogamy or heteronogamy. So the only option is xenogamy. Okay. So the case of papaya is easy to like, if you only want xenogamy to happen, you have to produce dioecious plants. So either the plant is going to be male or it's going to be female. Now, if I'm talking of how are you going to prevent autogamy? Produce unisexual flowers. But if you are producing unisexual flowers, like either it's a male flower or a female flower, but the male and female flowers, they're present on the same plant. That is, the plant is monoecious. Then you can prevent autogamy, but not heteronogamy, as we are observing in the case of castor and maize. Okay. Uh, So now we have completed the outbreeding devices part, which is given. Outbreeding devices essentially is equal to contrivances of cross-pollination, and it's a very, on-demand question for boards. You might be asked to write what are the strategies adopted by different plants to promote self-pollination or to promote cross-pollination. So in the cross-pollination points, you can add heteromorphism, you can add hergogamy, you can add dichogamy, you can add unisexuality, you can add the pollen pistil interaction, which include self-incompatibility, and it's promoting the growth. It's promoting the growth of pollen tube of non-self pollen. So these are all contrivances or strategies for cross-pollination, or you can say outbreeding devices. Now coming to the second topic that we were supposed to discuss, that is pollen pistil interaction. See, as I had already mentioned in the beginning of today's lecture, that the topic that we are going to study today, that essentially more of analytical topics, and we are primarily going to stick to NCERT, literally read through it, because all the scientific knowledge and understanding that you require has already been discussed in very detail in the past 4 lectures, and I request that you guys just go. Now, starting with pollen pistil interaction. The pollination, so reading the NCERT again, pollination does not guarantee the transfer of the right type of pollen. Okay, so now this is very interesting. See, I had just given you a reference of all these things in the previous 4 lectures. Okay, so what does it say? Pollination does not guarantee the transfer of the right type of pollen, compatible pollen of the same species as the stigma. Okay, so now think of it. For example, a particular honeybee is going to a particular flower collecting nectar. So while collecting nectar, the pollen grains are attached to all of its surface. Now it's going to another flower. Now the thing is that the second flower that it is going to, it need not be of the same species as the first flower, right? So the first time the honeybee can go to, say, flower X, like the flower is of species X. The next time it's going to a flower, it's of another species. So now this pollination is not going to happen, right? So now it's going to flower Y. Again, it's going to a third flower, which are the same species as that of the first. In that case, like the honeybee has pollen all over its body surface, right? Now the pollens of species X, they are going to be pollinated over the third flower, which is the species X, but not the pollens of species Y. Okay, so this is what it's saying. The pollination does not guarantee the transfer of the right type of pollen. That's why I'm always emphasizing on the fact that cross-pollination is not a surer process. It's a more complicated process due to the involvement of a third-party agent, the pollinator over here. So pollination does not guarantee the transfer of the right type of pollen, the compatible pollen. What does your compatible pollen mean? Your compatible pollen means pollen of the same species as the stigma. Okay. Often pollen of the wrong type, either of other species or from the self plant, if it is self-incompatible, also land on the stigma. Okay, I have already discussed that. It might happen the pollen of some other species is falling on the surface because suppose it is being carried by wind. The wind doesn't know, right? So the wind can't differentiate that this is a pollen of this plant, this is a pollen of this plant of this species. Okay, it has no idea. It's just doing its work of pollinator. Okay, so there is no distinguish between the pollens which are being carried. So there is a higher chance that the pollen of the different species can fall onto the stigmatic surface of different species. Or if in case the stigma is expressing self-incompatibility, then the pollen of the same plant, that is the self pollen, is also not acceptable on stigmatic surface, right? So the stigma is pertaining to the strategy of self-incompatibility. In that case, even if it's a self-pollen, it's the self-pollen is of the same species, right? But due to it being a self-pollen and the stigmatic surface pertaining to self-incompatibility, it's also not acceptable. So now, next, what is happening? The pistil has the ability to recognize the pollen, whether it is of the right type that is compatible or of the wrong type that is incompatible. If it is of the right type, the pistil accepts the pollen and promotes post-pollination events that leads to fertilization. So here is the thing. See, I'm telling you, this part is really fun. It's really, really interesting. It's completely analytical. If you just go, like you have studied all the technicalities, you go through NCERT once, the concepts are going to be crystal clear to you. It's really interesting. If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between the pollen grain and the pistil, mediated by chemical components of the pollen reacting with those of the pistil. It is only in the recent years that botanists have been able to identify some of the pollen and pistil components and the interactions leading to recognition followed by acceptance or rejection. So what is happening? So there is some sort of a chemical dialogue between the pollen and stigmatic surface. So the chemical components of the pollen. So like I'm talking of a chemical dialogue happening between the pollen grain and the stigmatic surface. So how are they conversing? Like they can't talk right as right. So you require something that is how they are conversing, the medium through which they're conversing. It's the chemical components of the pollen grain. So the chemical components of the pollen grain, they are reacting with the stigmatic surface. As a result, the first step is recognition. The stigmatic surface of the pistil is recognizing the pollen. Okay. So you have to keep this in mind as well. Like it has been in the very recent times, the botanists have been able to identify some of the pollen and pistil components. Okay. So we have got like research is still going on and we are gradually learning more about this chemical dialogue. So now you can think of it, right? So think of it in this way. So you are going down the road. Okay. You meet a person. Okay. You meet a person, you talk to the person. Upon talking to the person, two things can happen. Like like, for example, you're walking down the road, you come across somebody you had met a long time ago, then you converse with each other, you talk, you have a dialogue, then you recognize the person. Oh, okay, I have met this person in this and that event. This is how I know him or her. Okay. Now it can happen two things. Acceptance or rejection. Acceptance means you're interested in conversing with the person more. You want like, okay, so perhaps let's grab a cup of coffee. And it can also happen that no, you're not interested in spending time with the person. Okay, maybe he or she had done something bad to you, or you don't like the person. That's why you don't want to spend time with that person. So this is rejection. So this is exactly what is happening in this case. So the pollination is happening. The pollen grain is falling on the stigmatic surface. Then there is this pollen pistil interaction. That is essentially a chemical dialogue between the pollen grain and the stigmatic surface. So there is a dialogue between them. So how is this dialogue mediated? It is mediated by the chemical components of the pollen grain. So after the dialogue, there is recognition. The stigmatic surface of the pistil recognizes the pollen. Now what can happen? There is either acceptance or rejection. So it's followed by acceptance or rejection. So how can it, so the cases in which it can be rejected if it's a pollen of a very different species. Okay, if it's a pollen of a different species, then it can be rejected. Or in cases that the stigmatic surface is self-incompatible, then the self pollens are also going to be rejected. Okay, so now, and in other cases when the pollen is compatible, so there is going to be acceptance. So compatible pollens are going to be accepted, compatible pollen, and the incompatible pollen are going to be rejected. Okay, I hope you understand what incompatible pollen means. So for example, we are talking of species X, the stigmatic surface of species X, and for example, a pollen of species A, B, C, Z, any other species falls on the stigmatic surface. This is a pollen of a different species altogether. This is not going to be accepted. This is incompatible pollen. Okay. Now, in case of as I'm repeating, in case, for example, the stigmatic surface is self-incompatible, then the self pollens are also incompatible pollens, so they are going to be rejected. In other cases where the pollen is compatible, it's going to be accepted. Okay. So now what happens next? Accepted, in case of accepted pollens, what happens? The pollens which are accepted, the compatible pollens, in that cases, we observe that the growth of the pollen tube, the pollen germination followed by the growth of the pollen tube in the style is promoted. Because the pollen is germinating, the pollen tube is growing through the style. This growth is promoted, okay, when the pollen is compatible. But when the pollen is not compatible, pollen germination as well as pollen tube growth is inhibited by the plant. Okay. This is essentially the pollen crystal interaction. So acceptance is followed by pollen germination and pollen tube growth for compatible and accepted pollens, and the rejected pollens inhibited. I'm writing in short, inhibited. What is inhibited? Pollen germination and pollen tube growth through the style is inhibited. So this is the total of pollen pistil interaction is happening. So you understand. So first is the pre-pollination events, or you can say pre-fertilization event, the development of the male and female sex organ. Then is pollination, the transfer of pollen grains from anther to stigma. Okay. Then there is this pollen pistil interaction which determines whether the pollen is compatible for the stigmatic surface or not. If in case the pollen is compatible, then the post-pollination events are promoted. That is pollen germination and pollen tube growth, they are promoted. Then it's followed by fertilization. Then it's followed by post-fertilization. See, this is a so detailed and so well-coordinated process. Okay. Like so beautiful, isn't it? Like so interesting. Like each and every single step of this whole process is so well coordinated and well decorated. So next is, as mentioned earlier, following compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores. Okay. So if you remember the structure of pollen grains, I'm drawing it in like, very short. Okay. There's some kind of this structure. You can refer to lecture 1 for more details. Okay. So there was the outer pollen, XII, intine. Okay. The XII and intine, they were made up of, not intine, sorry. XII was made up of sporopollenin. Okay. So you had, let's just draw in rough. You had XII, you had intine. The XII was the outer layer, intine, the inner layer. The XII was made up of sporopollenin, etc., etc. And they were the germ pores. Okay, I've covered all this in detail in the first lecture. Okay. So what is happening? As mentioned earlier, the following compatible pollination. So now pollen pistil interaction is over. A compatible pollen has fallen onto the stigmatic surface. Now what happens? The pollen grain germinates on the stigma, pollen germination, and produces a pollen tip through one of the germ pores. So what happens? So this is, the stigmatic surface. The pollen grain is falling over here. Consider this structure. Now pollen germination is happening. The pollen tube is, see, this is one of the germ pores. Through this, the pollen tube. This is pollen germination. Okay. This is a germ pore. So this is the pollen tube is starting to grow. The contents of the pollen grain move into the pollen tube. Okay. So this is, the style, ovary and ovule. Okay. The pollen tube goes to the tissues of the stigma style and reach the ovary. So very clearly, so obviously there's going to be different cells and tissues out here, right? So this pollen tube is growing through the cells and tissues and reaching the ovule through the ovary. Okay. You would recall that in some plants, the pollen grains are shed in the two-celled condition, a vegetative and a generative cell. In such plants, the generative cell divides and forms two male gametes during the growth of the pollen tube in the stigma. In plants which shed pollen in the three-celled condition, pollen tubes carry two male gametes from the very beginning. So the pollen tube, after reaching the ovary, enters the ovule through the micropyle. Okay. So let's discuss the first part. I had discussed, okay, I'm telling you, I had discussed most of these topics in the previous lectures. This is just, these two topics are very analytical. Just, clear your mind and listen to what's being discussed. Okay. So, just a second. So we had before pollination, we had two kinds of pollen grains. In 60% of the cases, we observed that the pollen grains were being pollinated in the two-cell condition. So we had a vegetative cell and a generative cell. You have to remember that the division was happening to asymmetric spindle, etc., etc. Generative cell was small, vegetative cell was bigger, and all this stuff. Okay. So in 60% of the cases, we are observing that the pollen grains are being pollinated in the two-cell stage, one vegetative cell and one generative cell. In the remaining 40% cases, the pollen grains were being pollinated in the 3-cell stage. One vegetative cell and the generative cell was dividing to form 2 male gametes. Okay, so now the pollen grain is falling onto the stigmatic surface. Pollen germination is happening. Via pollen germination, the contents of the pollen grain are moving to the pollen tube, and this pollen tube is growing through stigma, style, and through ovary, it's reaching the ovule. Now, as it reaches the ovule, during this time, so let me write this. During the growth of the pollen tube to stigma, let me just take the line of the NCRT. The generative cell divides into 2 male gametes. So for whom does it occur? For I'm writing it as a 60% case. So where the pollen was being pollinated in the 2-cell condition. In them, what is happening? When the pollen tube is growing through the stigma, the generative cell is dividing into the 2 male gametes. In the remaining 40% cases, what are we observing? That during pollination only we had 2 male gametes already formed. So there is nothing to worry about. Okay. So now what's happening? The pollen tube is growing through the stigma, style, ovary, and reaching the ovule. And then what do you observe? That there was a vegetative cell, a generative cell, pollen tube formation is happening. And the generative cell is like during its growth through the stigma, the generative cell is divided into 2 male gametes in the 60% case. In the 40% case, we already have the 2 male gametes. So now the male gametes are moving through the pollen tube. Now next, so the after pollen tube reaching the ovary, it enters the ovule through the micropyle and then enters one of the synergies through the phylipum apparatus. Okay, so I think I have discussed this. There was porogamy, mesogamy, chalazogamy. Okay, I think I've discussed. Okay, let me just check one of the previous slides. Give me a second. Just give me a second. Let me check one of the previous slides. Okay, perhaps I not discussed. So maybe I'll just discuss this in short. Okay. Yep, no, I haven't discussed. So let me just discuss this in short. Now, this is not much of a big topic. It's a fairly short topic of discussion. Okay, so you have to remember this structure of the structure might be bad. Okay, so now you have to remember. Okay, so now the pollen tube growth, it can so this is the pore, right? So it happens. So this is essentially the embryo sac, where you are going to find the developed, mature embryo sac, in that 7-cell and 8-nucleate condition. So the pollen tube, what can happen? The pollen tube can come like this and get into the embryo sac like this. So like this is the micropylar end. This is ME, micropylar end. This is the chalazal end, CE. So what can happen? The pollen tube can enter after ovary, it's entering the ovule through the micropyle as well as the embryo sac through the micropyle. So the pollen tube is entering the ovule through the micropyle. See, like this is like this is structure of ovule, right? We have discussed this in detail. Okay, of integument, chalaza, hilum, funicle, everything. We have discussed all this. Okay, so the pollen tube is entering the embryo sac as well as ovule through the micropyle only. Okay, this is one case. In the other case, what are we observing? It can happen that the pollen tube is entering through the chalaza and then it's coming like this and it's entering the embryo sac like this. So it's happening that it's entering the ovule through the chalaza, but it's entering the embryo sac through micropyle only. Why are we saying like only micropyle is the way it's entering? Because you see this whole embryo sac is covered by double layered integument on like the whole, like if you go to the structure of ovule, we will observe that like the integument is covering the whole of your ovule like this. Only in the micropyle it's open. So the pollen tube has only a single option to enter the embryo sac, that is through the micropyle. But for ovule, it has 3 options. It can enter through micropyle, it can get in through chalaza, or it can get in through one of the integuments. 3 options: micropyle, chalaza, or one of the integuments. Okay. Most probably, I think I've discussed this before. So, okay. In case I have not, I have just discussed it right now. So, if we are having 3 options, why, like The pollen tube has three options to enter the ovule. One through the micropyle, second through the chalaza, and third is through the integument. But to enter the mature embryo sac, where essentially your antipodal cells, synergids, ovum, or like even double nucleus, all are present over there, right? To enter the embryo sac, it is only a single option, that is the micropyle, because it's covered by double layered integument on the both on like across all its sides. Okay. Like remember why I'm saying double layer, you will observe that in the diagram there is outer integument and inner integument. Okay, mind that. So that's why for the pollen tube to enter the embryo sac, we have only a single option, that is the micropyle. So the pollen tube after reaching the ovary enters the ovule through the micropyle. Like this is the majority, but chalazogamy, porogamy, mesogamy, all three are there. Okay, so that's it. So the next we are moving on to many recent studies have shown that the filiform apparatus present at the micropylar end of the synergids guide the entry of the pollen tube. All these events from the pollen deposition on the stigmatic surface until the pollen tube enters the ovule are together referred to as the pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeder in manipulating pollen pistil interaction, even in incompatible pollinations to get desired hybrids. So now see, this was essentially the concluding line for pollen pistil interaction. So see, pollen pistil interaction is a vast process. So after pollination is happening, the pollen grain has fallen onto the stigmatic surface. Then there is a chemical dialogue between the pollen and the stigmatic surface mediated by the chemical components of the pollen grain. Okay, according that there is recognition, then it is acceptance or rejection. The accepted ones are the compatible pollens, the rejected ones are the incompatible pollens. The incompatible pollens, in them pollen generation and growth of pollen tube is prevented, is inhibited, and in case of the compatible pollens, the pollen germination, the growth of the pollen tube is favored. So now the pollen tube is growing. So when the pollen tube is growing through the stigma, in that case, we are talking at the 60% case where the pollen grains were pollinated in the two-cell condition. While the growth of the pollen tube through the stigma, we are observing that the generative cell is dividing into two male gametes. In the remaining 40% cases where the pollen grain was being pollinated in the three-cell condition, there's nothing to worry. We already have the two male gametes. So now the pollen tube is growing through the stigma style ovary and then coming to ovule. In ovule, we are observing that it has three ways to get into the ovule: through the micropyle, okay, then through the chalaza, and then through the integuments. Okay, so when it's getting in through the micropyle, we have this is porogamy. When it's entering through the chalaza, it's chalazogamy. When it's entering through the integuments, it's mesogamy. I've discussed all this. Next, but when we are talking of entering of the embryo sac, then there's only one option: through the micropyle. The pollen tube enters the mature embryo sac only through micropyle. The reason being it's covered by double layered integument on all sides except at the micropyle. So to enter the ovule, you have three options. To enter the embryo sac, we have only a single option. And upon entering of the this whole total process, starting from the pollen deposition on the stigma until the pollen tubes enter the ovule, is together referred to as the pollen pistil interaction, which is a very, very dynamic process. So now, why is the knowledge of pollen pistil interaction important? It has been mentioned in the last line. It says, if you have a proper knowledge of this pollen pistil interaction, you can manipulate this process and cause pollination even in incompatible pollens, resulting in the development of desired hybrids. So let me just write this: importance of pollen pistil interaction. So the importance of pollen pistil interaction is so if we have proper knowledge of the pollen pistil interaction, we can manipulate that and bring about incompatible pollination. So now what does incompatible pollination mean? So if the pollen that had fallen onto the stigmatic surface was an incompatible one, then in that case, the pollination wouldn't, the pollen pistil interaction would not proceed, right? But if we can manipulate that, causing about this kind of pollination, which is being referred to as incompatible pollination, then we can get to obtain desired hybrids. So this is the importance of pollen crystal interaction. Okay, so in it, like it's a very interesting topic. A lot of questions can come from this topic on your NISER exam, IISER exam, NEET exam, and in boards also it can come as a direct question, like describe the process of pollen crystal interaction. So do keep the importance in mind. Okay, so why is our knowledge of pollen crystal interaction important? Okay. So you can easily study the pollen germination by dusting some pollen from flowers, just pea, chickpea. Okay. So this is essentially a lab part. So you guys are fortunate enough that you would be able to experience biology lab in class 11th and 12th. We were the COVID batch. So in class 11, at least, I personally didn't get the opportunity to attend labs, but in 12 I did, and it's really fun. So this is your one of your lab assignments. The next is, as you shall learn in the chapter on plant breeding, that is chapter 9, a breeder is interested in crossing different species and obtain genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization is one of the major approaches of crop improvement program. So what is happening? So let me just put it in this way. For example, consider Marvel. Okay, so what are the ideal characters you can consider? Like think of a person who has the technical mindset, the technical knowledge, the technical expertise of Iron Man, has the leadership skills, the captain skills, the leadership qualities of Captain America, has the strength of Thor or Hulk, whoever you consider, has the agility of Black Widow, has the precision, the sharp shooting ability, as our what's his name? Arrow. Okay, the person who throws arrows. Okay. Okay. Oh, I just forgot his name. Anyhow, okay. So this is if we have such a character in Marvel, this is going to be the ideal case scenario, right? So you have technical expertise like Iron Man, you have leadership skills like Captain America, you have inhuman strength like Thor or Hulk, you have agility like Black Widow, you have very sharp shooting precision skills. Okay, like Hawkeye. Okay, Hawkeye. Okay, yeah, I just have not forgotten. Like you have your sharp shooting ability like Hawkeye. Like it's a combination of a list of desirable characters which makes you the best and the most competent of all, right? So when we are talking of plant breeding, we are interested in crossing different species such that the very highlighted desirable characters of each participating plant species, they are combined so that we are producing superior varieties. So like I gave the Marvel example, let's just put it in case of plant. So we have a number of species, and each species we have some particular desirable character. So we are combining all of them through artificial hybridization. Artificial hybridization is one of the major approaches for crop improvement program. Via artificial hybridization, we are combining so that we are able to produce superior varieties containing some desirable characters from all of this. Okay. In such crossing experiments, it is important to make sure that only the desired pollen grain are used for pollination and the stigma is protected from contamination. This is achieved by emasculation and bagging techniques. So, okay, so I think the pollen crystal interaction portion is complete. So I think this is going to be the last part of the topic that we're going to discuss, emasculation and bagging. Okay. So after emasculation and bagging, I think I'm going to wrap up for this lecture. In the next lecture, we are going to proceed to how the pollen tube enters the embryo sac. Double fertilization is very short because it's not take 5 years, and then to post fertilization events. So what is happening? So like in case of artificial hybridization experiment, okay, what is very important that it's accuracy. Okay, accuracy as in for example, I want the pollen of this particular. Okay, I think like if you guys have read genetic, you have basic idea of Mendel's experiment. You out of studied in class 9 and 10. So it's a basic idea of using Mendel's experiment as well. So what did we do? Like he used all of this in case of pea plants. So selected pollens were allowed to pollinate and fall onto the stigmatic surface. It was make sure that any sort of unwanted pollen, which is here is essentially contaminating the process. Here is like contamination. That is, any unwanted pollen is not falling onto the stigmatic surface. Mendel took great care of that. So in case of artificial hybridization also, this is very important that you out to take care so that only your desired pollen fall onto the stigmatic surface. So for this, two techniques are used. One first is bagging and emasculation. This is a very easy topic. Like, I think it is pretty evident from the name itself. Okay, so what is emasculation? If the female parent bears bisexual flowers, okay, removal of anthers from the female bud before the anther dehisces using a pair of forceps is necessary. This step is known as emasculation. So, like, very easy. Obviously, you can understand emasculation. That is, you are cutting off, you are removing the male reproductive part. This is emasculation. So in case we have bisexual flowers, we have a bisexual flower, we want to make it a unisexual female flower. So using a pair of forceps, we are removing the male reproductive whole, that is the androecium. This is the process of emasculation. Bisexual flower, we are turning it into a female unisexual flower, which is a female flower. Using a forceps, we are removing the male reproductive whole. This is emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of this stigma with unwanted pollen. This process is known as bagging. So here the compound you have to remember, that is butter paper. It is not compound actually. It is the substance that is used. Like butter paper is used for bagging. Like obviously it has respective chemical compositions. If you are interested, you can Google it, but it's not relevant in this case. Just knowing about butter paper would do. So what's happening? So now we had a bisexual flower. By using a pair of forceps, we removed the male reproductive part, the androecium. Now we have a female unisexual flower. So now to prevent it from contamination, that is, so that any unwanted pollen does not fall onto the stigmatic surface, we require to cover this flower, cover this emasculated flower. Okay, so how are we covering it? We're covering it with a bag of suitable size made up of butter paper to prevent contamination of this stigma with unwanted pollen, and this process is called as bagging. So when the stigma of a bagged flower attains receptivity, mature pollen grains collected from the anther of the male parent are dusted on the stigma, and the flowers are again rebagged and the fruits are allowed to develop. If the female parent produces unisexual flowers, there is no need of emasculation. The female flower buds are bagged before flowers open. When the stigma becomes receptive, the pollination is carried out using the desired pollen and the flowers are rebagged. Okay, so let me just explain this and then we will be good to go. So what is exactly happening? So like, think of it. You are like, consider like whatever I'm discussing now, you are going to find greater reference to this when you are going to be studying about Mendel's experiment. Okay, so you have, for example, you have a unisexual flower, unisexual male flower, unisexual female flower. So in that case, you don't require to emasculate, right? You have a female flower. So you have a female flower. What you are doing it? You are bagging it. That is, you are covering it with a bag of suitable size made up of butter paper. You have bagged it. Next, what you are doing? You are waiting for both this male plant, like you have bagged, so contamination is not possible. Now you are waiting for this female plant, female flower as well as male flower to mature and attain receptivity. Like for the stigma, it's receptivity. Okay. So once both of them are mature, you are taking the mature pollen grains, like brushes are you like in brushes can be used. So you are using brushes to collect the pollen grain from the anther of the male counterpart. And then you're opening the bag, you're dusting that on the stigmatic surface, and then you are again rebagging it. Why are you rebagging? So that you are preventing contamination. That is, so that the unwanted pollens, undesirable pollens are not falling. Okay, so this is the process that you're adopting. So in case you are having unisexual flowers, just bagging is enough. You bag the female flower. Once it attains receptivity and the pollens are mature, you brush the pollens and make them form a stigmatic surface, rebag it, over. But in case where you have bisexual flowers, you require, so to prevent like a self-pollination, you require to emasculate. Obviously, why are you emasculating? To prevent self-pollination, right? You are converting the bisexual flower into a female flower to prevent self-pollination. So using a pair of forceps, you're removing the male counterpart. Now you have a female flower. You had a bisexual flower. Now you're having a female flower. You're bagging it, waiting for the stigma surface to become receptive. The male counterpart, the pollen grains are maturing. Like repetition, brushing, making the first stigmatic surface, again rebagging. This is exactly what is happening. So bagging and emasculation, there are two techniques which are adopted, and we find extensive use of them in artificial hybridization experiment as well as they have a lot of on-field use as well. So in plant breeding techniques also, we find extensive use of these techniques, and also in the laboratory for research stuff. So, okay, so I think we have covered, firstly, we had covered outbreeding devices today, which is essential contrivances of cross-pollination. where we had unisexuality, dichogamy, halogamy, heterogamy, and a new thing that we discussed today was self-incompatibility. Next, we proceeded to pollen-pistil interaction, where we discussed about whether a pollen is compatible or not, then the development of the pollen tube through the stylostem ovary, how it enters through the ovule and then through the embryo sac. We discussed the total of pollen-pistil interaction. Okay. And next, we discussed about bagging and emasculation, which play a critical role in the artificial hybridization techniques. We discussed about the importance of pollen-pistil interaction. We discussed about the importance of hybridization. Okay, so now what we have, the male and so now the pollination has happened, pollen tube formation has happened. In both the cases of pollen grains, we have the male gametes. Now it has reached the embryo sac. So to the next class, where does the story lead to? The pollen tube is going to get into the embryo sac. The male gametes are going to be released. Double fertilization is going to happening. Then we are going to proceed to the post-fertilization events. I think in a lecture or two, this chapter will be over. So thank you guys. Thank you for listening to this lecture. My best wishes. Continue studying and my best wishes. And yeah, do take care of your health. Okay, in these times, do take care of your health. My best wishes are with you.